All right, hello everyone, and welcome back. So now we're going to be going into part B of the central nervous system. So in these parts, we're going to be, finishing up the brain and how you can form language, memory, and a little bit about sleep and consciousness. And then we're going to go into the spinal cord and talk about its anatomy and structure. So first step here is we're going to go over language. So how do we understand language? So this is a multiple association areas. So the main areas include that Broca's area and that Wernicke's area that I talked about in part one. Then we're talking about memory. So everyone wants to learn, how do we remember everything for the exam? Well, I'm going to tell you how it works. I've told you all how it works before, but you forgot it because you didn't do these key steps in memory formation. So I'm going to teach you the secrets of life and how we can memorize things. And then you're going to be mad when you realize it still requires work. Can't just take a magic pill and remember everything for the exam. And then we're going to talk about brain waves and EEGs, consciousness, and your sleep and wake cycle and how it's regulated in different parts of that sleep and wake cycle. So first here, language, again, the main areas include the Broca's area and the Wernicke's area. So Broca's is involved in speech production, and then Wernicke's is involved in understanding spoken and written words. So if you have a lesion in your Broca's area, you can understand words but cannot speak. If you have a lesion in your Wernicke's area, you can speak, but words are nonsensible. You require both areas here to work. And then corresponding areas on right side are then involved with nonverbal language components as well. Now, memory, this is all about storage and retrieving that information. So different kinds of memory. Declarative memory, this is memory of facts, so names, faces, words, dates, etc. So declaring a statement, think about it as that, or declaring a fact. Procedural memory, this is memory of skills, so it takes time to learn it. So playing a piano is an example of procedural memory. And then motor memory, this is something like riding a bike, driving a car, memory of motor skills. Walking is one of the simplest forms of motor memory. And then emotional memory, this is memory of experiences linked to an emotion. So when you hear a rattlesnake, your heart starts pounding. You're ready for that fight or flight response. So now there are two stages of declarative memory, and the one everyone here is focused on, how do we form long-term memory? How can I remember anatomy and physiology forever? Because it does have a limitless capacity. I don't know if the book was referencing the movie Limitless here, but that's how it talked about it, a limitless capacity. So you all have the ability to memorize anything and everything you want. And don't think of it as memorization. Think of it as learning and forming these associations. And then memory becomes easy then. Short-term memory, though, that's what you don't want to have when taking this course. You forget it when you go to sleep. This is limited to 7 or 8 pieces of information. So there is a limit to your short-term memory. And this is why we break up the lectures in these different parts to give your brain a break to reset things. And then this is why we do hands-on stuff as well, because then you can form associations with the facts. So then factors that affect the transfer of short-term to long-term, one is your emotional state. If you're happy, so alert, motivated, surprised, or aroused, you actually learn better and you can make that long-term memory. Rehearsal, this is called studying. Repetition and practice. And then association. So this is where we form multiple association between vision, hearing, writing. This is why writing notes is so important. You're writing it down and then you're reading it. And then you're touching the models and then you're listening to me. You're watching these videos. It all links to this emotional memory. Then automatic memory, the subconscious information is stored in this long-term memory. All right, so then how do we turn this, how do we put this all in an image? So how outside stimuli are all these associations coming in. So vision, touch. Why this touch is a needle coming into the finger, I don't know, but it could think of this as touching the model. You're listening to me. You're not really smelling me, but don't worry about that. But it's another association you can make for studying, smell. So let's say you're eating a delicious pizza as you're studying. You can smell that pizza, and then you think about how long-term memory is formed. So yeah, let's use that one for smell. So these are all the afferent inputs coming in, and this all creates a temporary storage in the cortex. And then if you don't ever access it again, you lose that data. But let's make long-term memory or short-term memory. So here, one pathway to get to long-term memory is this automatic memory. And that's what everyone's trying to figure out. And not as much is known about transferring this outside stimulus directly to automatic memory. Usually it takes a very strong stimulus for this to happen. But here, the data is selected for transfer then. This becomes momentarily short-term memory, and then you typically forget it and it's lost. Now let's imagine we studied. And you were happy when you were studying. So excitement, rehearsal, association with new senses. This then causes long-term memory formation. But if you don't continue to practice and restudy, you could lose that as well. So this is all communicating with each other, and the ultimate goal is to form that long-term memory, of course. Then clinically here, if you damage your hippocampus or the temporal lobe, may result in only slight memory loss, but bilateral destruction can cause widespread amnesia. So there are two main types of amnesia. Interograde amnesia is when consolidated memories are not lost, but new inputs are not associated with old ones. Think anterior, so new forward memories are not associated with the old ones. So the person lives in here and now. So the memory of a conversation just 5 minutes before would not be remembered. This is like Dory from Finding Nemo. So she remembers other things in a past life, but the recent conversation, she forgets who you were just a little bit later. So that's interograde amnesia. And then retrograde amnesia is loss of memories formed in the distant past. So retro video games are old video games in the past. I'm getting old enough where now retro is considered old. But see, this is loss of memories formed in the distant past. So if I had retrograde amnesia, I would no longer remember retro video games, sadly. So now, depending on the situation, you might need to get an EEG. An EEG is an electroencephalogram. So this records electrical activity that accompanies brain function. So if your brain is being studied for certain patterns, you would get one of these EEGs. So these are used for diagnosing epilepsy and sleep disorders, and then local lesions, tumors, infarcts, or infections or abscesses can be diagnosed via these EEGs as well. It's also used in research and also to determine brain death. We're going to talk about brain death a little bit later. So here, how this works, you can see all these electrodes out here on this individual. So electrodes are placed on the scalp, and they measure the electrical potential between various cortical areas. So different things we talked about, epilepsy, and so here talking about seizures a little bit. So an epileptic seizure is a torrent of electrical discharges by groups of neurons. So it's these neurons firing in rapid repetition. So victims of epilepsy may lose consciousness, fall stiffly, and have uncontrollable jerking. And there are different forms to regulate epilepsy, and it varies from individual to individual. Like the keto diet, lowering the amount of glucose getting to the brain is one of the ways epilepsy can be treated. So what could happen before seizures is neurons could start firing. So this aura could happen, which is a sensory hallucination which may precede a seizure. Absent seizures, also known as petit mal, these are mild seizures of young children where the expression goes blank for a few seconds. Then tonic or clonic seizures are also known as grand mal. These ones are the ones that are very violent, and the most severe could last a few minutes. Consciousness, they fall down, bones are broken during intense convulsions, loss of bowel and bladder control, and severe biting of the tongue. So if someone's ever having a seizure, you should never have your finger near their mouth. So this control of epilepsy includes anticonvulsive drugs, vagus nerve stimulator. We'll talk about the vagus nerve later this semester, or deep brain stimulator implantations that deliver pulses to the vagus nerve or directly to the brain to stabilize brain activity. So the big issue with these seizures is that uncontrollable neuron activation in the brain. And then next topic here is consciousness. I'm not going to talk too much about this, but this is just perception of sensation and just multiple integration. So mental processing, so memory, logic, judgment, etc. is consciousness. So it's clinically defined on a continuum that grades behavior in response to stimuli such as alertness, drowsiness, stupor, and coma. And then the sleep-wake cycle. The book goes into lots of detail on the sleep-wake cycle. I'm just talking about it a little bit and the importance of sleep. So the two main types of sleep, again, you get these sleep patterns via an EEG. There are apps on your phone that you can sit on your bed that are trying to analyze your sleep, but if you have anyone else in that bed with you, a pet or a person, it's not going to do a proper analysis. But the stages of sleep, there's non-rapid eye movement, which is NREM. That's broken down into 4 stages. Again, we're not going to break that down. And then there's rapid eye movement, or REM sleep. So the importance of sleep, this is when your brain is flushing itself out. So there are so many studies that go into the importance of sleep. Big one is your immune system. So they tested individuals who were sleep deprived versus not sleep deprived post-vaccination, and they found that individuals that were not sleep deprived were able to form the antibodies faster after the vaccination, whereas the people who were sleep deprived, it took them longer to build up that antibiotic, or that, not the antibiotic, but that antibody memory, and that it's called your titer, or titer, T-I-T-E-R. So that's the concentration of antibodies in your system that you build after getting a vaccine to perform that memory response. And we'll talk about the immune system later this semester. But they found that sleep deprivation had a major effect on that. So here, slow wave presumed to be a restorative stage, and people deprived of REM sleep become moody and depressed. So all these different stages are important. So the sleep may give the brain an opportunity to analyze the day's events, work through emotional events or problems, or eliminate unneeded synapses that were formed. So you literally dream to forget. So then clinically, here are some other things that can happen that lead to loss of consciousness or brain death. So fainting or syncope. This is a brief loss of consciousness, so most often due to inadequate cerebral flow. So let's say you stand up too fast, so your blood pressure severely drops too low, and you pass out. So due to low blood pressure or ischemia from hemorrhage or sudden severe emotional stress. And then coma. Now, we've all heard of coma before. This is unconsciousness for an extended period of time. So it's not the same as deep sleep. So here, oxygen consumption is lowered in these conditions, whereas your oxygen consumption during sleep is still the same. And then brain death is irreversible coma. And then we're not going to really get into it, but it has all the ethical and legal issues that surround decisions on whether the person is considered dead or alive if they're deemed brain dead. Okay, now the brain needs to be protected. So it's sitting in these layers, or also known as these meninges. So they cover and protect the central nervous system. So we have blood vessels going through here, cerebral spinal fluid, and also forming the different partitions we have in the skull. So there are 3 layers. From external to internal, we have the dura mater. So right here, we have the dura mater. It's made up of 2 layers here, the periosteal layer and the meningeal layer. So this is talking about the brain right now. And then we have the arachnoid. Arachnoid is spider-like. So right here, we have the arachnoid matter, which is this layer right here. And we see these little fibers in here. And these fibers are where the arachnoid matter gets its name. Think of it as like these little, cushions or springs in the brain. And then the pia matter is the connective tissue that directly covers the brain. So these different layers are very, very important in protecting the brain. And then we have these little granulations that could pop out that connect to these sinuses, which are storage areas for blood. And then looking at the dura mater here, so this is an actual, dissection over here of a human skull showing these layers and these how it goes around. So up here, so this fibrous connective tissue, again, this is the strongest meninx. The periosteal layer attaches to the inner surface of the skull, found only in brain. So this one is not in the spinal cord. The meningeal layer is then in the spinal cord and extends into the vertebral canal as spinal dura mater. Then when we go over the spinal cord, we'll see this one come back. These layers are mostly fused, but in certain areas they form those venous sinuses, which collect venous blood from the brain and empty into the jugular veins of the neck. And then the pia mater is that delicate connective tissue right on the brain there. So following every convolution of the brain contains many tiny blood vessels that then feed the brain. You can also see these fibers going through here for the associations in the white matter, which are kind of cool. And then cerebral spinal fluid, as I talked about in part A, this gives the buoyancy to the brain. It's floating in the cerebral spinal fluid and protects the central nerve. So here it actually reduces the weight of the brain by 97%. So the brain will crush under its own weight. And so here, the cerebrospinal fluid is formed by choroid plexuses. So there's a choroid plexus down here by the cerebellum, and there's one up here by the thalamus, hypothalamus. So I just want to switch to this picture here. So here it's formed by these choroid plexuses, and then it just follows a pathway around the brain. So here, just traveling around, it flowing outwards through these arachnoid granulations, traveling around, and all the way down the spinal cord. So this same cerebral spinal fluid that's in the brain is also down the spinal cord. This is important later when we talk about when they test your cerebral spinal fluid. They don't take it from your brain, they can take it from the lower end of your spinal cord. And then we talked about these cells in last chapter, these ependymal cells. Ependymal cells, their purpose in the brain, they're these ciliated cells that move the cerebral spinal fluid around. So here we finally get to see their importance. So this is showing what that choroid plexus does and how it produces cerebral spinal fluid from the interstitial fluid of the blood. So one thing that can happen with the cerebral spinal fluid, if it's blocked, if you can't drain it, it could begin to build up and increase pressure. In newborns, so this is called hydrocephalus. In newborn, skull bones are unfused, so the pressure causes the head to enlarge and you can drain it. But in adults, this could be very dangerous because the skull bones are fused, so what happens, the pressure just builds up in the brain. Remember, P1V1 equals P2V2, and if you increase the pressure, then what happens? So it will increase the volume of this fluid, the pressure increases, and you don't want to increase the pressure on a delicate tissue like the brain. So this can compress blood vessels and crush the soft nervous tissue. So treatment to drain CSF with a ventricular shunt to the abdominal cavity is very important here. And then, as we talked about in the last chapter, the astrocytes are those cells that form the blood-brain barrier. They wrap around the capillaries and control what comes in and out of the brain. So it helps maintain that stable environment. And the brain has its own immune response. The microglia that respond in the brain, they don't have the normal immune response in there because you don't want high levels of inflammation in the brain. So here, these are exceptionally impermeable tight junctions to keep the brain separate from many blood-borne substances. This blood-brain barrier sometimes might receive abbreviated as the BBB in papers. So here it's a continuous endothelium of the capillary walls, and it has a thick basal lamina around the capillaries and the feet of the astrocytes and surround the neuron. So it controls what comes in and what goes out. And then here is also showing it. So here's the capillary. Here are these little endothelial cells around the capillary with these tight junctions. And then here's a basement membrane around that. I know we haven't talked about the vascular system yet, but this is common of the vascular system for the capillary bed to have this pericyte. So that's common right there for what it looks like. And then also we have this extra layer with these astrocyte feet wrapping around it. Again, that is controlling what comes in and out of the brain. So it allows certain nutrients to move by facilitated diffusion. Metabolic waste can get in and out, and allows any fat-soluble substance, so including alcohol. This is why alcohol has an effect on your brain, nicotine, and anesthetics as well. And if any new drug design is happening and they have to get through the blood-brain barrier, that's usually the main preventative thing that occurs in drug design. So when something works amazingly well in a petri dish, and then they realize this drug won't pass the blood-brain barrier, that becomes a big issue, because then if it's a drug that's supposed to have an effect in the brain, it could be a problem then. So this is absent in such areas though. So like the vomiting center, where we need to monitor the chemicals coming in and out. So it's monitoring the chemical composition and temperature of blood in certain regions. So it's a little loose in certain areas. Now, a couple degenerative brain disorders. The one I studied for my dissertation was Alzheimer's disease. So if anyone wants to talk more about this, feel free. I know all about it. So this is the one that results in dementia, memory loss, disorientation, and so forth. And it deals with this protein called beta amyloid. These chunks, think of them as stones, form in the brain. And there's still a lot of debate on what is the toxic component or what kills the neurons in this. So those form outside the cell. Inside the cell, these other things form called neurofibrillary tangles from this protein called tau. So you're getting these stones that are forming inside the cell and outside the cell. And it's just a very severe disease that eventually kills the neuron. So we can see extreme brain shrinkage here as the brain cells die. So still a lot of debate on Alzheimer's disease to what is actually the degenerative mechanism behind it. And then Parkinson's disease, we talked about this one earlier when talking about the basal nuclei and how it causes the shakiness. So what happens here is it causes a loss of dopaminergic neurons in the substantia nigra. So what this is dopamine isn't important for regulating this stuff. So causes unknown, but theories include mitochondrial abnormalities or protein degradation pathways. And there's a protein involved here called alpha-synuclein as well. Treatment includes L-DOPA, so trying to mimic dopamine, deep brain stimulation, and gene therapy. And then Huntington's disease, this is a pretty severe disease, and it is inheritable. And it's very odd because it doesn't present itself until later in life after the individual could reproduce. And it's a dominantly inherited disorder. So if that person had children, they know there's a 50% chance that their child could have it as well. And then that child sees their parent go through Huntington's disease, which is a pretty severe disease. So usually fatal within 15 years of onset. And, it's just a slow mental deterioration. There's not a lot of treatment. So treatment is with drugs that block dopamine effects. And symptoms are wild, jerky, flapping movements. And stem cell implant research is promising, though. But it causes a severe enlargement of the ventricles over time. And like I said, it is genetic. And it's a dominantly inherited disorder. And then to go through a little bit more here, we need to talk about the spinal cord. It is part of the central nervous system. So anything outside of the spinal cord is then peripheral nervous system. Now, one thing a lot of people don't realize, the spinal cord actually ends at the L1 or L2 vertebrae. So right here. Then it separates into different nerves. So this spinal cord, of course, provides a two-way communication to and from the brain and to the body. As we talk about the spinal cord, peripheral nervous system, autonomic nervous system through the next couple chapters, we're going to be talking about the spinal cord and how things move through it quite a lot. Also, it's a major reflex arc center. We're going to talk about reflexes coming up later as well. So the spinal dura mater is one layer thick. It just has that meningeal layer. Then it also has this epidural space, which is a cushion of fat and a network of veins in the space between the vertebrae and the spinal dura mater. I'll show that on an image coming up later. And then this also fills, the cerebral spinal fluid fills this as well. So between L1 and L2 then is the site of a lumbar puncture. So right at the end of that, because they could get the cerebral spinal fluid from there. You don't want to take the cerebral spinal fluid from the brain if doing a spinal tap, because you could cause some really, really severe damage if you screw up the needle when in the brain. Whereas if you screw up the needle down here, it doesn't affect as many nerves, but it's still dangerous. So then looking at the different layers here, there's a cervical enlargement in the neck region. So here's the thoracic spinal nerves, the cervical spinal nerves, and the lumbar spinal nerves. So just referring, there are 31 pairs of spinal nerves here, and those spinal nerves will come up again next chapter. And then here's that dura and arachnoid matter that we find there. In the lumbar region, there's something called the lumbar enlargement, and then the spinal cord ends right here at L1, L2. That's when it becomes what's called the conus medullaris. So it's just this cone-shaped structure. That's right there. And after the conus medullaris, it becomes my favorite part, the cauda equina, which is a collection of nerve roots at the inferior end of the vertebral canal. So all these nerve roots then. And it literally gets its name. It means horse's tail. So if you look at what this region looks like, it kind of looks like a horse's tail if you do a dissection. And then it terminates down here at the filum terminal, where the sacral spinal nerves are. And all those sacral foramens, all those holes, are exits for the spinal nerves in the hip region. So then this is showing a lumbar punction. It's actually showing it between L4 and L5. I won't ask you the direct spot, but this is just showing another example. There's still cerebral spinal fluid down here that you can take it from. So this is just showing an example of that, different ligaments around it and so forth. So right here, it's just getting that cerebral spinal. This is the same cerebral spinal fluid that is found in the brain. Okay, now we talk about the spinal cord and a cross section of the spinal cord. Make sure in class you look carefully at these models. Similar models. There's a model that looks just like this one and a model that looks just like this one. And they all have these different parts, so make sure you can name all of these parts. So I'm going to switch to the pen here when highlighting some of these parts. The key here is we have information coming in right here. So this is a spinal nerve. We have information coming in. This bulge is called the dorsal root ganglion. So right here is the dorsal root ganglion. And that's one way you can orient yourself when you look at a model of the spinal cord. Look for the dorsal root ganglion, or the DRG. That is on the dorsal side. So then you know the difference between dorsal, so anterior versus posterior, or dorsal versus ventral. For the spinal cord, we refer to things as dorsal and ventral. So here, this is the input or afferent pathway. So somatic sensory input is coming through here, and it enters this part of the spinal cord. This part of the spinal cord, let's see, where is it labeled? But it's called the dorsal horn. I think it's labeled on one of the next images here. But I'm going over it all here. So right there we have the dorsal horn. Then this is the gray matter in here. So it forms a little interneuron. It could go over the gray commissure here. So the gray commissure. Oh, right here, dorsal horn is labeled. Right here's a dorsal horn labeling on this side right there. Dorsal horn. So right here's the central canal for blood vessels going up and down, and cerebral spinal fluid in there. And then this is the gray commissure if it needs to cross over to the other side. So they can go over here, and the output has two choices. If it's autonomic, typically it comes out not the ventral, but the lateral horn. So right here is the lateral horn. We're going to talk about the lateral horn more when we get, when we get to the autonomic nervous system. And then down here, we have the ventral horn. Another way to orient yourself on the spinal cord, there's this ventral median fissure, which is a pretty deep fissure on the actual spinal cord. You can see it on this one right up here. So that's on the ventral side or the anterior side. So here is the ventral one, and this is where the signal then comes out. So for somatic motor output, I'll show that a little bit more later as well. And then some more of a cross-section up here. Here's that epidural space. This is that fat layer. And the model will show blood vessels in here as well. So make sure you check those out so you can identify that fat layer. Then you have the subdural and subarachnoid space where you have the cerebral spinal fluid. And then you have the dura mater and pia mater in there as well. So here you have the input. It can go through the integration center here and then come out for the output or efferent pathway. So yeah, so this ventral root is coming out, and then the dorsal root comes in. And we'll be talking about that more later as well. So make sure you can identify all these different parts of the spinal cord on the spinal cord models. Very, very important. Here is talking about the dorsal, ventral, and lateral horns. I have a nice image that shows these coming up later. Just know that these are gray matter, and they're part of these interneurons here. And we'll talk about these more on the next figure, but I was just wanted to highlight where they were on the first one. So they're there, and there. And there's one on the other side as well. So these lateral horns are also only in the thoracic and superior lumbar region. So these are part of the sympathetic nervous system, which makes sense because it's autonomic. So sympathetic nervous system, autonomic. We'll talk about that more when we get to the autonomic nervous system. That gray commissure is the bridge that connects left and right, and it's gray matter, and it encloses the central canal. And then the ventral roots, dorsal roots, and the dorsal root ganglia. These are all input information. So it's dorsal root ganglion. A ganglion, so just so we have the nomenclature down, a ganglion is a cluster of cell bodies in the peripheral nervous system. A nuclei is a cluster of cell bodies in the central nervous system. And then after that, we become spinal nerves, which are next chapter. So now when we break down those horns, this is how they break down. And you guys have this image to fill in your workbooks too. So here, this is the somatic sensory for blue. or SS, so somatic sensory. So it's your muscles, your tendons, your ligaments, the sensory input from them coming in. So right here we see this bulge. So right here would be the afferent pathway. So then it hits an interneuron right there, and same thing with all the sensory input from your organs. So visceral sensory is here as well, or VS. So here it hits an interneuron, showing all these little interneurons in here. And then it could go different ways. Here, visceral motor. So anything that's autonomic is going to be a visceral motor output. You don't have to tell yourself to digest your food. Your body handles it automatically. So VM is visceral motor, and SM is somatic motor. This is what you are controlling. So these come out that ventral horn. Visceral motor go out the lateral horn, but they still then become part of this ventral root as it exits then. So if you can understand this figure, you're then understanding which part of that gray matter handle somatic sensory, visceral motor, or somatic motor. And then the spinal cord has all this white matter in it as well. So remember, white matter, this is long communication network. So we need to send signals to the brain. So that's, of course, we need to have these ascending tracks. So this goes to up to the higher center. So when you're walking through a dark room at night and you step on a Lego, you realize that's a Lego, but you initially jump off. So you have a reflex arc that goes through your interneuron and back out. But then you also have these ascending tracks that then go up and form associations where you know you just stepped on Legos. So you have to send that input to your brain, so you have long distance travel for that. So these ascending tracks are here in blue. I'm not going to expect you to memorize each of these different regions here, but these are just, if you see white matter, it's long distance. So here the dorsal side is mostly ascending. So these fasciculus gracilis, fasciculus cuneatus are all ascending tracks. They just go to different parts. So there's these spinocerebellar tracks that are also going up as well, and these spinothalamic tracks. So they kind of tell you, so to the cerebrum or to the thalamus. And then descending tracks, so corticospinal tracks are your pyramidal tracks that we talked about for your primary motor cortex. So these descending ones are in red here. So red pen isn't a good color. And they just are, connected, not connected, but just highlighted in red here. Then there's some other ones: rubrospinal tract, reticulospinal tracts, vestibulospinal tract, and tectospinal tract. Again, you don't have to know what all of these do. Just understand that white matter has ascending and descending tracks as part of it. And here, just keep going through here and just showing a couple different examples here. So here we're getting input from a muscle spindle, and this is going up the spinocerebellar tract, which is fine-tuning and getting that input from your muscles for the tension, so you can fine-tune that output. But just going through that interneuron here, and it goes up that ascending track, goes all the way up, and then it integrates into the association area in the cerebral cortex. And then here it's showing the dorsal column medial lemniscal pathway. This is just a touch receptor like on your foot and a joint stretch receptor on your hand, and just showing how it can integrate up different pathways here, whether it's the fasciculus cuneatus or fasciculus gracilis. So here it jumps through different interneurons, and here you notice how it jumps to the other side of the brain as well. So what started on this side jumped to the other side. And then this is just showing a descending pathway, so showing the pyramidal track starting up here in the the primary motor cortex, sending that signal out these projection tracks down through the matter, and here they jump over and then switch sides, and then here, so they're controlling the opposite sides for wherever they're going. So it's just two different examples here of how these ascending and descending tracks work. Understand that there are hundreds of thousands of these tracks. So then there are some different conditions that can happen with the spinal cord. We know about some of these. So localized injury to the spinal cord or its roots lead to functional losses. So paresthesias is caused by damage to the dorsal root. So imagine if you damage that dorsal root, you're no longer getting sensory input. So you have sensory function loss, but you still have motor output. If you step on a Lego, you don't know you stepped on anything. And then paralysis, damage to the ventral root. So if you damage those ventral roots, you have no motor output. So it leads to either flaccid or spastic paralysis. Flaccid paralysis is when you have severe damage to that ventral root, and so impulses do not reach muscles, so muscles atrophy. And spastic is when upper motor neurons or the primary motor cortex are damaged. So you can't control output. It's only reflexes that are still working. So muscles are stimulated by reflex activity. So you step on a Lego, you jump off that Lego, but you can't tell yourself to control that output. It's only reflexes that are working. So there's no voluntary control of muscles. Very interesting. And muscles often shorten permanently because of this. And then some different spinal cord traumas that could occur. So different transections or cuttings of the spinal cord depend on which level it was transected at. So paraplegia is transection between T1 and L1, and quadriplegia is transection in the cervical region. So no appendage use. And spinal shock is a transient period of functional loss to the caudal, lost caudal to the lesion. So it depends on where it was. And then poliomyelitis. So polio is an infective disease, thankfully eradicated. So destruction of the ventral horn motor neurons by the polio virus. So if you're destroying the ventral horn motor neurons, you're destroying that motor output. So muscles atrophy, and death may occur from paralysis of the respiratory muscles or cardiac arrest. Survivors often develop that post-polio syndrome many years later from that neuron loss, where they never fully regain that muscle output. And then ALS, or Lou Gehrig's disease, amyotrophic lateral sclerosis. So this is an autoimmune disease that causes destruction of the ventral horn motor neurons. So again, if you're destroying the ventral horn motor neurons, you don't control output. So this causes a loss of the ability to speak, swallow, and breathe, and death typically occurs within 5 years. A very, very debilitative and progressing disease caused by environmental factors, genetic mutations involving RNA processing, and glutamate exocyticity. And some drug can interfere with that glutamate signaling, and it's the only treatment right now. And it deals with the destruction of those motor neurons. So last slide here, and the last 3 things I want to talk about. One is cerebral palsy. Cerebral palsy. This is a neuromuscular disability involving poorly controlled or paralyzed voluntary muscles. So you can't control that motor output. And this could be due to brain damage, lack of oxygen during birth could be a reason for this, where you don't get full development of that, or you get damage to that primary motor cortex. So speech difficulties, motor impairments can be seen. Some patients have seizures, intellectually impaired, or are deaf. And visual impairment is common in this as well. And then anencephaly is when the cerebrum and parts of the brainstem never develop. So imagine if you never develop the brainstem, where all these important regulatory functions are taking place. So the neural fluid fails to fuse, and unfortunately the child is vegetative, and the death occurs soon after birth. And the last one I wanted to mention is spina bifida, where you get an incomplete formation of the vertebral arches. So it doesn't fold properly, and the spinal cord could actually pop out a little bit. So most cases are caused by a lack of B vitamin folic acid. So B vitamin is really important during development, and U.S. incidence has dropped with mandatory supplementation. All right, so that is part B. And again, if you have any questions, feel free to email me and let me know. And I hope you all have a great day, and see you all in class. Bye-bye.